Okay, here we go. So we're back for the second session. In this session, we're going to talk more about life history development. So just a few words to introduce you to the jury members and reviewers of this session. So in the jury, we have Kayla Plichon and Paola Furla, like for the first session, joined by Aldi Daniel from IRCAN, Université Côte d'Azur, and also joining you for the reviewing, so Kayla, Paola, also Anne Pajot, Eric Rottinger, and myself. Those are people in this group. As usual, 15 minutes presentation, around 20-minute questions. You're welcome really to formulate your question at the end. And if you're ready, the floor is yours.
What if a coral species is highly susceptible to lethal disease? Should we still attempt to restore it on site? This is the question I'm going to address today by assessing the feasibility of in situ restoration for the groove brain coral, Diploria labyrinthiformis, under pressure from stony coral tissue loss disease, Sciroldi, in Curacao, by looking at early stage survival, tissue loss progression, and early growth as indicators. So for a bit of context, for the past 5 decades, disease outbreaks have emerged as a major stressor in the Caribbean Sea, leading to destructive declines of 50 to 80% in coral cover. Together with climate-driven disturbances and anthropogenic pressures, disease are further weakening the resilience of Caribbean reefs and also hinder restoration efforts. In 2014, a new disease emerged in Florida called stony coral tissue loss disease, or Sciroldi for short. It has rapidly spread throughout the Caribbean and led to severe regional declines in both reef condition and cover. Sciroldi has been described as the most persistent and widespread coral disease documented to date, and it's now affecting over 22 species of massive, slow-growing, reef-building corals. Among these, the groove brain coral, Diploria labyrinthiformis, or DLAB for short, is categorized as highly susceptible to Sciroldi, and also critically endangered on the IUCN Red List. In wild colonies, the disease causes rapid lesion progression of up to 15% tissue loss per week, causing high mortality rate. And in 2023, the disease reached the island of Curacao in the Southern Caribbean, where the local NGO Branch Coral Foundation quickly observed the dramatic effects of the disease on mainly old and large DLAB colonies. So this is a healthy colony in June, and a few months later, it succumbed completely to the disease. And I myself saw it as completely dead and covered in algae in April this year. Yet, disease impacts and the disease dynamics and the impacts on Curaçao's reef have not been studied yet, and no restoration initiative was so far suggested to restore the affected species around the island. But in the wake of the disease front, active restoration of Sciroldi susceptible species is critical. However, there's a major knowledge gap on the feasibility of in situ restoration under disease pressure. The studies so far have studied Sciroldi in dynamics in wild colonies, therefore we don't know much about the nursery fragments responses to disease exposure. So this leads me to my research question. Is in situ restoration of Diploria labyrinthiformis within a Sciroldi affected site feasible? To guide us in our investigation, we will look at 3 subcategories of response variables, evaluating the early stage survival. Then we will analyze the progression of tissue loss. We will then characterize the early tissue growth and also identify the factors that influence these responses in microfragments. We are doing this on a very short timescale of 10 weeks. Therefore, this study is mainly to guide future restoration efforts, and will not look into the deep coral physiology or immunology. How did we do this? So we're here in Porto Mori Bay on the south side of Curacao, and this is a site with high prevalence of Sciroldi. Within this site, we chose a point 100 meters from shore where there's already a successful staghorn coral nursery to place our 2 nursery trees. These nursery trees are here at 8 meters depth, and in this area is where we collected our donor colonies. So we chose donor colonies that didn't have visible signs of bleaching or Sciroldi lesions. In these colonies were then fragmented into 199 fragments, and into 4 size classes to test these effects of the different size classes. We also have mounted them on different substrates to test these effects, so ceramic plates and plastic plates that are commonly used in restoration. Then we also tested for the effect of the position of the fragments in the tree with top level and bottom level fragments. However, due to field constraints, colonies 1 and 2, all their fragments were placed on the top of the trees, while all the fragments from colonies 3 and 4 were placed on the bottom of the tree. So this does not allow separation in the statistical analysis, which I'll explain later. So we then looked at 3 response variables to assess the feasibility of restoration. First, the percentage of tissue loss and the progression rate. So in week 0, I calculated the surface area of each individual fragment, and then analyzed every week after that if they showed tissue loss. I estimated the percentage using image J. Then the second response is survival, so it also relies on tissue loss. So with a threshold of 60% tissue loss, fragments were considered dead. This is a limitation, however, it's justified by the absence of tissue regeneration over the repeated visits, and also the rapid algae overgrowth of the bare skeleton, which together suggests irreversible necrosis and unlikely recovery of the fragment. The third response variable was early tissue growth. So it was calculated as the difference in live tissue surface area between week 10 and week 0. This is an example of a fragment that showed a slight positive increase in tissue. Then these 3 response variables were assessed against the 4 experimental factors that I explained to see which factor influences the response the most. So let's look at our results. So overall survival was 37.8% with 73 fragments alive in week 10. This is low survival, but let's look at the differences in colonies. So we see striking differences with colony 2 fragments showing the highest survival of 95.8% at the final week, with followed by colony 1, which showed 66.2% survival, but colonies 3 and 4 showed very low survival of 6% and 0% respectively. This was supported by a Cox proportional hazard model that showed a significant effect of colony origin on fragment survival. So this test shows that by taking colony 1 as a reference, we have a lower mortality hazard for colony 2 fragments and a higher mortality hazard for colonies 3 and 4. This is indicated by the hazard ratio value here. Then we also see overall a pattern in terms of the temporal phases. So we have, for the first 5 weeks, a sharp decline in survival, followed by a more stabilization here, except for colony 2 that had pretty much no death. But in the outbreak phase, so this really distinguishes 2 different phases, outbreak phase with 109 deaths recorded in total, and then in the stabilization phase, we have 11 deaths. And we see also this differences in phases in the tissue loss progression rates. So in the outbreak phase, we have a rate of tissue loss of 15.9% per week, compared to a rate that's much lower in the following weeks of 1.4% per week. Then what affects these, what influences this tissue loss susceptibility? Well, we have found a colony origin effect with colony 1 and 2 having lower tissue loss overall compared to colonies 3 and 4 in all the 3 weeks that we tested. So I tested the week 3 to represent the outbreak phase, week 5 to represent the median survival week that is in between these 2 phases, and week 8 to represent the stabilization phase. And Driscoll-Wallace and post-hoc done tests showed significant differences over all 3 weeks. We have a similar results for the position in the tree, with fragments on top having lower tissue loss compared to those on the bottom in all 3 weeks tested. However, again, the experimental design does not allow separation between colony and position effects, and there might be some confounding effects here. Plate type and size classes only had a significant effect on tissue loss in week 3, and after that it was not significant anymore. As you can see here, plastic had a higher tissue loss than ceramic, but then it was not significant after the following weeks. In terms of growth, there was no significant difference between the final and the initial live tissue surface area of the fragments. However, we have 29 of the 73 surviving fragments that showed a slight positive increase in tissue, indicating some growth. And this again was influenced by a colony effect, with colony 2 fragments showing a positive change in live tissue, significantly different from colony 1 that showed a slight negative growth. I can explain later if you have questions. I know negative growth can be confusing. But, all fragments were this time on the top of the trees, since we only had colonies 1 and 2 remaining alive. So there is no confounding effect to be considered here. And we found no significant differences in plate types or size classes. Now, what influences this high mortality peak in the outbreak phase of 54.7%? Well, other studies on boulder coral restoration have found a similar high early mortality, possibly caused by handling, mechanical damage, and fish predation. And here we had a quite heavy handling and wounding caused by microfragmentation of the corals that could have increased stress and disease susceptibility by allocating their resources more in rapid wound healing instead of immune function. So this could explain it, but also we attributed the lesions to Skittle D. How did we do this? Based on 3 aspects. So first, the similarity of the wounds to those reported in the literature. It's often described, Skittle D lesions are often described with a sharp boundary between the live tissue and the bare skeleton, which was observed here. Also, the tissue loss progression rate was similar to that reported in the literature, with our outbreak phase tissue loss progression of 15.9% compared to wild colonies that showed 15.5% tissue loss per week in Florida. And lastly, the high prevalence of the disease in Porto Mori Bay in our study site increases the probability of transmission of the pathogens to the fragments. However, no biopsy was done here, so we should not claim 100% confidence on Skittle D being the cause of mortality. So future studies should take tissue samples to analyze the microbiome of these colonies and fragments. Then what influences the decrease in tissue loss rate and mortality rates in the later phase, so in the last few weeks? Well, this indicates that some fragments might show more resistance than others to early mortality spike. And this, we identified that was maybe not so much influenced by type class and plate type, but more, the main drivers were more colony origin and position that are the main drivers of resistance, early stage survival and growth. However, again, confounding effect, and we could not fit a mixed effect model to test these possible interactions due to non-normality of the data. However, we can discuss these different effects qualitatively. So the position in the tree has been suggested as potentially leading to higher tissue loss and mortality in nursery settings due to reduced light availability, increased sedimentation, and pathogen introduction. However, studies suggest that in nursery settings, fragments responses in wound healing, disease resistance, and survival and growth are more strongly influenced by colony-specific genetic variations rather than by nursery design or environmental factors. So this suggests that colonies 1 and 2 were maybe more resistant genotypes, or maybe had holobiont communities that were beneficial to disease resistance. However, again, no genotyping was done of the donor colonies, so future studies are encouraged to do genotyping to investigate this further. In terms of implications for future studies, so this really emphasizes the need to monitor wild colonies to identify resilient or resistant donor colonies and to improve restoration outcomes in the future, so prior to restoration, but also to continue long-term monitoring to do comparisons with the wild to see what factors influence performance, but also with control in land-based nursery settings. Monitoring environmental parameters is also crucial. This was done, but not comprehensively here. And future studies are encouraged to improve design and replicates across all colonies and treatments to draw more robust conclusions. Finally, using preventive and mitigation measures to minimize the skittle-D impacts on nursery fragments could also be useful in these settings. To go back to our research question, is in situ restoration of the Floria labyrinthiformis within a skittle-D affected reef feasible? Well, we have results that indicate that despite high early mortality, a subset of the fragments survived, resisted disease pressure, and showed early tissue growth. Therefore, in situ restoration under disease pressure is feasible if resilient donor colonies are selected. Further optimization, however, is needed to improve protocols for donor colony selection, microfragmentation, and monitoring specifically to restore these species where they are declining. Because ultimately, protecting and restoring coral reefs is an ecological and a socioeconomic imperative. Thank you.
Okay, here we go. We can start with you, maybe Aydin.
Yeah, so thank you. Thanks for this really nice talk, really well structured. So do they have any idea about what, I mean, the origin of this disease? Is it, do they think about the way it spread? So any idea about that?
It cut off a little bit, but I think you mentioned how it spreads and what the pathogen is.
Yeah,
So the pathogen is actually not confirmed yet. There's been studies, I think I have something here, but so the pathogen is not described yet. They have, however, looked into how it spreads. So it's mainly just through the water column and through direct contact. In this case, the fragments were close to each other, so there was a possibility of transmission between the fragments. That's why we also have this 60% threshold where we remove the fragments. I didn't mention that, but we considered them dead at 60% because we also had to just remove them from the nursery to avoid transmission. This was based on recommendations from the literature. So yeah, we have mostly characterized how it looks, but there's still some uncertainties on the pathogen itself and the transmission, especially in nursery settings. It's more complex.
Okay, then I have so regarding the coral that get infected, or that got this disease, so is there a size effect? Do you know if the biggest one
in the wild
more sensitive? Yeah, into the wild.
Yeah, in the wild, it's definitely affecting more the older and bigger colonies, as I mentioned in my introduction. It's mainly the big ones, which is even more concerning. But also the recruits have been found to be affected a little bit. It's still in the early stage.
Okay, thank you. And last question. At one point on your slide, I think 42, you make a comparison between the different colony, and as references, you are taking the colony 1. So I would like to understand, why did you take this one as a references to compare to the other and not the one that have the highest survival?
Yes. So actually, this is more of a statistical testing requirement that I was trying the Cox regression models and didn't know that you needed to take a reference. So it just automatically took the first one on top of the row. And then I thought of rerunning it with the colony 2. However, it takes a lot of time, and also I don't know if it would have changed much, because in the end, colony 1 is the one in the middle, so it's good to kind of see, I don't know, the difference in the two extremes.
Okay, it's the one that survived the most, if I understood well.
No, colony 1 is the one 66%.
Yeah, so it will change completely half of the conclusion. But yeah, okay, thanks a lot.
We can move to Paola and with Kayla.
Hi again. Thank you very much for your presentation with a lot of work, a lot of nice results that you've got. I have some questions about your experimental design and how this could influence your conclusion. So you put your colonies on these kind of trees of transplantation, some on the bottom, some of on the top. So do you know what kind of differences? Okay, sure, the light, but is there any other differences on the situation on the environment of those colonies in the trees? And on the other way around, do you think that transplantation in trees represent really what's going on the field? And for this particular species, is the good, what kind of other kind of transplantation you can do on restoration as kind of experimental design?
Okay, your first question was how the position, mainly you want to understand the position effect?
Yeah, you say top and bottom. We don't know exactly the distance between the top and the bottom, and what kind of differences you can have. Is there only light constraint, light criteria that can change, or there could be also other environmental factor that can influence?
Yeah, so we have top and bottom. The nursery was placed at 8 meters depth with a rope of 1 meter. So that means the bottom level was probably at 6 meters depth, but the top level was just above, like maybe 1 meter difference between the two. And the light availability was really decreased by the fact that we put both arms on top of each other instead of en quinconce. So this is definitely an improvement to do. I didn't have time to mention it, but they were really on top of each other. Then I also mentioned, I think that bottom fragments have been studied to increase higher sedimentation from the ground. And also in our study site, we had a flood in week 3, I think, which could have influenced the results by a runoff of fresh water, which could have increased sedimentation and impact more the bottom fragments than the top ones. Yeah. And then your second question.
Well, the second question, yeah, you closed my answer to my second question, but the idea is when you put the colony in those trees, on those restoration devices, you isolated them from the rest of the biodiversity. And do you think that this could have an impact on the sensitivity and fragility of the colony from bacteria or virus? Because I don't think that it's clear this white bind disease is due to bacterial viruses.
Yeah, so it's not sure yet, but it is definitely a concern. That's why I wanted to see if it's really worth putting them in such high amount of stress. So microfragmentation is really known to cause a high degree of stress, and putting them back in that same reef where there's the disease is a high risk. However, there's also different methods that have been suggested as colony, what is it called? Colony fusion, microfragment colony fusion, where you can place the fragments really, instead of putting them, I place them on separate plastic plates and separate plugs. You can also put them directly in the field, or even maybe in nursery settings, very close to each other to fuse, to encourage fusion, because they will grow towards each other to form back to cover the wound. I'm not sure if that answers a bit your question.
Yeah, I thought about biodiversity in general in the reef, because the bacterial sink could be in different organisms, not only on cnidarians, and the fact that you isolated the colony one to each other, but also from the reefs, because you isolated the colony from the reefs, that could enhance the fragility of the corals.
Yeah.
Because there is no more friendly microbiome that can get and help to fight against the pathogens. So this is more what perhaps to isolate the colony in those trees will not reflect the robustness of the colony in the field.
Yeah, definitely. So that's why we could also, I know some studies have done direct transplantation or outplantation of after microfragmenting the colonies, putting the microfragments directly on the reef. However, they saw a high increase in algae overgrowth in those cases because it's much closer to the ground. And but there might be benefits of interactions with other colonies. However, the other colonies might be infected with Skittle D and therefore ease the transmission as well.
Colony or other, even other
species,
other species. Yeah.
Yeah.
I have a second question, and it will be my last one for you, so I can give the floor to Kayla.
Okay, and the question, I misunderstood. Sorry, go ahead, Paola.
You say that the genotype selection could be a good way to accelerate or to have a better restoration, but how you can apply genotype identification and in which kind of genome target point you will focus in order to select those genotype?
Yeah, so this links back to Emily's presentation, I think, a little bit. We talked about how can we use this information for restoration. So in this case, I think it was specifically in Diploria labyrinthiformis, they identified specific endosymbionts that were more resistant to Skittle D. So we could use some genotyping and then holobiome manipulation. I run through it a little bit in my conclusion. I didn't have much time, but holobiome configuration or modification has been suggested as a pre-restoration method. However, this may be not scalable for small NGOs, and it's more research, I think, maybe not as realistic for restoration in small scale, but it's definitely used. Could use some research.
Thank you.
Yeah.
Congrats on your presentation, and I'm going to jump on Paula's question about the genotype. So you said you couldn't genotype your colonies, but of course, again, NGO context, it's understandable. However, are you sure you're comparing the same species? Are you sure the colonies are from the same species?
Yes, I'm pretty sure. No, because there are like two Diploria colony species, and I want to go back to a picture of nice colonies. They are very distinguishable. In the first few weeks, actually, I got trained by my supervisor to recognize them in the field because they have this Diploria labyrinthiformis, so it forms a labyrinth, and you can really see in these turns, there's a small crevice, and this really distinguishes them from other species. In my annex again, I'm left, but I have pictures of other species that could use the restoration because they are also affected. Here. So this is the other Pseudodiploria species, and it doesn't have this crevice. It's more one.
And do you know if there is any cryptic species within your species?
That I don't know.
That couldn't be a possibility why you have such difference in the response.
It could be, yeah.
So are you sure about the species you're having?
Well, then in this case, no, but cryptic species, they haven't been described. At least there's not been one described.
I have a protocol question. So you did the fragmentation of the colonies, and how much time did you wait before deploying the trees? Did you allow the fragments to heal a little bit before, or were they directly put again in the site?
They were directly put again in the site. So you deployed the trees without the fragments first, a few weeks before, just so that it was less time for them to be spending out of the water when we would be ready to do the microfragmentation. And when we did microfragmentation, took the colonia out, put it straight in a bucket of water in the shade, and did the microfragmentation maybe as fast as we could in an hour and then deployed them back in.
And don't you think that creating this fragmentation and wounds in the fragments, putting them back in a site that is already contaminated with this disease, could also potentially increase your death rate?
Yes, yes.
What would you suggest to do differently?
Yeah, so also in my conclusion, don't have time to go through it, but they suggest doing antibiotic baths before putting them back in the reef. Sorry?
How long?
I'm not sure because it's not been researched yet. It's also NGO level that are trying it in actually in the other island of Bonaire. That's the neighboring island. There's an NGO that is doing trials with antibiotic baths.
Don't you think it could be a possibility to go to another site that is not contaminated, let them heal, and then put them back into the sites?
Yes,
Would that be feasible?
yeah. I was wondering why are we putting them back where they are declining, but it's also to restore the reef that is declining. There are other
If
reefs.
you're letting them, let's say, 2 weeks
Yes.
to heal, and then to put it back to
Putting them in
a pristine reef,
offshore site, but pristine.
for example.
Yes, that could work. However, there's very little reefs that are pristine in Curaçao left. But in general, putting them there and then bringing them back would also, I don't know if you would need to bring them back out of the water to deploy them again. So I think that would increase stress as well.
And you think the stress will be less in that case than putting them into an immunity stress because they have to heal and fight this contamination they have?
Yes, I think could be an option to look into.
It's okay. I think those were my. Oh yeah, I have another question. So you said that you remove fragments that had 60% loss of tissue, and you consider them as dead. Did you follow them afterwards to see if they actually were dying, or if you placed them in a tank or whatever, and to see if they could recover? Or what did you do with these fragments that you removed?
We removed them and we did not have facilities to observe them afterwards.
What did you do with the fragments?
Probably we put them in a bucket and put bleach and not my decision. See that full recovery.
Do you think they could recover? Because 60% tissue loss is still 40% of tissue present, right?
So,
what do you know about the capacity of regeneration of these?
Recovery has been described as very unlikely when
in this species.
In this species.
Because also some depending on species, you can have this variability between species and how they recover and how they react to everything.
Yes.
So, do you think it could be a possibility to? If you put them back in a stable environment, they could recover, and you're not losing these fragments. In a restoration setup, you want to have the more material, the better, right?
Yeah. I didn't look specifically in the recovery of the species in the wild because I think it might not be applicable to fragments because of that stress that was caused from microfragmentation. So I looked more into the possibility of recovery in microfragments of other species as well, which is very low when as soon as you see necrosis with algae overgrowth, the disease is, I observed it as just continuing and being uninterrupted until full death. However, I had a few fragments that showed slight tissue loss, and then it stalled, and those ones I kept them in the nursery site. But I didn't observe recovery in the sense that it didn't grow back, or it's also very short scale, so maybe now they grew a little bit more tissue on the sides, but not where the algae overgrew the dead skeleton.
And do you have updates of on how your fragments are doing?
I've been trying to get updates, but it's hard to communicate with a small scale NGO. They are continuing the monitoring, but not as not really. I sent them my protocol. I made a new protocol and I sent it to them. I'm not sure they're following it because I got pictures of the whole nursery trees as a whole, but he didn't continue the actual individual pictures and analysis. But someone to take over. So Yanni will look after my babies. We are still doing it. Like the rate after those 10 weeks of mortality hasn't changed. I think maybe one fragment died more, but it really stabilized it, and now they have those fragments and they're pretty stable.
Good. So we can open the floor for a couple of questions from you guys. Someone is interested on the back.
Did you notice if the original colonies were affected by the disease? Like the
ones I collected?
Yeah, the ones that you fragmented from, like the ones I'm assuming you left some.
Some of them were some.
The original colonies, or did you take the whole colony to fragment?
No, yeah, we took the whole colony because they were pretty small, and we got between 25 and 70 fragments out of each. But no, yeah, they were selected because they showed no visible sign. Again, we cannot be 100% sure that they didn't have like a, how do you say, when you are in incubation of the disease and you don't show signs yet, that we're not sure, but they were selected because they were pretty intact with no discoloration or disease lesions. And the disease lesions would be very distinguishable, especially in wild colonies. You would see a spot of white, actually bare skeleton.
You mentioned at one point negative growth. What does that?
Of course, we come back to that.
I mentioned I could explain.
Yeah.
I'm sure there is a slide on there.
Where was it? So yeah, the negative growth is a result of microalgae overgrowth on the exposed skeleton edge of the fragment. So here we have not consistently removed the excess skeleton from the fragment. So it is a recommendation in the literature. However, as I mentioned, the microfragmentation procedure was very rapid. So sometimes this is a bare skeleton, like excess skeleton that we should have removed, that led to much higher algae overgrowth. This is taken in the same week. And this is a flat fragment that showed, I think, growth, whereas this one might be overgrown by algae and therefore shrinking in its tissue over time. This is an example as well. I didn't have the comparison with week 0, but it was definitely like this was live tissue growth. This was live tissue, and now it's kind of shrinking in size because of algae overgrowth.
Yes, Amy.
I was curious so you said that genomic analysis would be interesting to be able to identify colonies that would be good to transplant for the future or do restoration efforts on. And I know that would be super hard probably for small NGOs to pay for that. Do you have any ideas of visual cues that might indicate if a colony is healthier or resistant? Maybe there's nothing, but I was just curious.
Well, that's actually what was hard to. I was the one selecting the colonies, and I was like, oh, which one should I? And it's just the visual. You just make sure there's no lesions or discoloration. But actually, the one I selected, colony 2, that was the one with highest survival. It didn't look the best. It was very, it was not very round. It was a bit imperfect, and it had a tube worm tube. And therefore, I was like, maybe this one's not good. But it was the one that had that showed the highest survival. So, not sure about visual cues. But we are trying to set up a collaboration with a lab on Curaçao as well and set up our own, not my NGO, but NGO is trying to set up a land-based nursery as well to be able to do these types of analysis prior to restoration.
Thank you. We have to go. Maybe the discussion can continue after. Congratulations again.
I love my NGO.
And so we will go for the last presentation of this morning. with Lisa Lafond.
It's super broken. Oh.
We are going to talk still about development, but larval developments of urchins.
So hi everyone, thank you for coming today, and I'm going to present to you the work I've been conducting within the Solamare research platform in Corsica, which is focused on aquaculture, as well as with the Aquabios project, which is an European project working on valorization of local resource and also sustainable aquaculture. And today the work I'm going to present to you is where I studied the effect of different microalgal diets on the larval development of Paracentrotus lividus. So as you might know, today a lot of marine resources are under pressure, and the Mediterranean isn't an exception. And in that ecosystem, we find a key ecological player, which is Paracentrotus lividus, the sea urchin you can see here, which has an important role in keeping the stability in rocky reef habitats. It's grazing on algae and prevents the overspreading. But sadly, today the wild stock are declining. As I mentioned, I was in Corsica, and on the island, the sea urchin has really important cultural and economic value. People enjoy them in what we call sinat, so it's eating your sea urchin by the sea with a glass of wine. It's really, really nice. But because the wild stock are declining, it's impacting the fishing and the fisheries. In 2024, they had to reduce the fishing season from 4 months to only 2, and in 2025, they had to even close it earlier, it at only 1.5 months in. That has a direct impact on fishermen. So one possible solution that arises is aquaculture that could have restored the wild stock while also supplying the market. And the wild stock is action that the lab is already doing, but supplying the market now. But one limitation to this is that the sea urchin larval phase is really fragile and is highly sensitive to its diet, because it has a lot of importance. And today, actually, they mostly rely on species that are coming from collection and that are not directly from the place where it is. That creates a need for a resilient microalgae that is local and really adapted to the sea urchin diets. If we go just a bit back to the sea urchin biology, so you have a bit more detail about what I'm going to talk to you about after. So it's an echinoderm that has a planktonic life cycle first, with the Puteus larvae you can see here. And then these larvae grow with 4 different stages, so the 4-arm stage, the 6-arm stage, the 8-arm stage, and the rudiment stage. And during this larval phase, they exclusively rely on microalgae for their diet, which is really important for their nutrition and also their metamorphosis before becoming benthic juveniles. And if we go back to the algae we wanted to investigate here, we wanted to try Chetoceros tenissimus, which is a diatom that was isolated from the Diana lagoon. So here in Corsica, my lab was just a bit more up, so it was really close from it. And lagoon strain of microalgae, they are known to have like advantage because they are in really fluctuating environment. The pH, the salinity, they are changing all the time. So they are more resilient and they have nutritional advantage. So that leads us to our research question, which was, can the local strain of Chetoceros tenissimus support the larval development of the sea urchin Paracentrotus lividus as efficiently as the standard hatchery diets today? And we did that evaluating its performance while looking at the survival, the development speed, and the growth of the larvae. So if we look at the experimental design, we did 2 different experiments, and it's important that right now I tell you that we did not plan to compare them. They're really different, and I'm going to present to you all of the results, but we can't compare the 2 experiments. So we tried 5 different microalgae: Chetoceros calcitrans, Chetoceros tenissimus, Pleuropellis sp, Rhodomonas stamina, and Phyllocylum tricornutum, and the 2 first are diatoms. This was an atophyte, cryptophyte, and another diatom here. And we had 9 different diets, so monoalgal diet with only 1 algae, bialgal diet with 2 different algae, and trialgal diet with different algae. And for each condition, we had 3 replicates. So maybe that's a bit more clear. So we have all of our monoalgal diets, only one, and our control, which was the 3-algal diet, and it's the diet they usually use today in the hatchery, in the lab. And we have the bialgal diet, so we have Pleurocrisis combined with Chetoceros calcitrans and Tenuissimus, and same for Nastalina, the first and the second diapan, and once again, the control. And we decided to end the experiments once the larvae, they reached 75%, the one we sampled, reached at least 75% of the last larval stage, which was the rudiment stage. And for the rearing conditions, so we set up 80-liter tanks in a room at 90 degrees, and we check the pH and the O2 daily, as well as we did density count. So we sampled water and we counted the larvae, and we also identified their stage to follow the stage they were at. And each time they changed stage, we took a morphometric measurement. So we took the total length of the larvae, the post-oral length of the arm, the body length without the arm, and the body weight. And we also looked at ingestion rates with the multisizer and lipid sampling, but these are ongoing analysis, so I'm not going to present to you the result of this one today. So if we move on to our results, so here we looked at larval mortality. We did this first one to be sure that all of our experiments were going well and that all the tanks were growing. So we have our monoalgal diets. Here you have the mortality percentage, and here you are going to have the algae treatments. We fed the larvae with, but we are talking about larvae all along the results. So here we didn't observe a significant difference with the tests between the diets. However, even if the difference might not have been strong enough, we can see that Chetoceros tenuissimus, so our algae of interest, and the hatchery control were the one that seemed a bit lower, but no significant difference observed. And if we move on to our bialgal diet, so we have the same organization. We can once again see that we did not observe any statistical difference, and the one that seemed to have a lower mortality was the hatchery control. But what we can take from this result is that all of the diet and the treatments reached the rudiment stage, so did the whole experiment well, and we didn't lose any tanks, so nothing crashed. And then we also looked at the development speed, so it's the time in days it took for the larvae to reach the 75% of the rudiment stage, so the end of the experiments. And here it's not exactly organized as just before, but it's organized as who finished first and who finished last. So you have the time in days, and you can see here that Chetoceros tenuissimus was the one that finished first in only 15 days, so it was really fast. And then the control that is here finished in 16 days, so it was 1 day more, and the one that took the most time was Chetoceros calcitrans in 20 days. So yes, Chetoceros tenuissimus did better than the hatchery control here. And for the bialgal diet, we have the same organization. And you can see here that Rhodomona salina with Chetoceros tenuissimus and Pleurocrisis sp with Chetoceros tenuissimus, so both with our algae, were the one that finished the fastest in 14 and 15 days. The control finished in 16 days, and the last one in 19 days. So here we can see that the one that included our algae finished first, and also, yeah, they did better than the control, but so yes, we observed difference. Everyone finished between day 14 and day 20, and the one that had Chetoceros tenuissimus finished. And finally, that leads us to our final result. So we looked at morphometric measurements. So that's the larvae at the 8 rudiment, like 8 arm rudiment stage. You can see the rudiments here. And that we look at the total length, the post-oral arm length, the body length, and the body width. And we did this morphometric measurement for each stage and each treatment. But today I'm going to present to you mostly the total length, because it's often a measure that is used as a proxy, and that it reflected what we observed in the other one. So I chose this one, so it's going to be more simple. So we looked at the 8-arm rudiment stage larvae, and here you have the size in micrometers and the treatment. And we can see here with the test that all of these diets did the same, and this one was smaller, so it was Ketoceros calcitrans. But we can see that Ketoceros tenuissimus, one algae that we wanted to test, did the same, like was statistically the same as the archery control. So that was a good thing. And for the biological diet, same, once again, the measures and the treatments, we could see that there was a statistical difference between two diets, otherwise the other they did the same. And it was Pleurocaris sp with Ketoceros tenuissimus and Ersalina with Ketoceros tenuissimus. So the two diets that included the algae, but they were statistically different from each other, but they were comparable to the archery control. But even if they were different, they did as well as the archery control. So what we can take from here is that we observed difference between the diets, but overall, the one that we were interested in matched the archery. So that leads us back to our research question, which was, can this algae support the larval development of Parasaurolophus libidus? And we had key findings that went that way. We had a stable larval development. We also had a fast development and an early rudiments formation, a good growth, as you could see. So everything matched the archery control. So that implied that suggests maybe that this algae, from what we saw here, has a high nutritional quality. And if we look at what stayed in the literature and we could compare to our study, they usually say that like monoalgal diets, so with only one algae, they are not really suitable to bring the larvae all the way to the rudiment stage, just before metamorphosis. But here, surprisingly, all of our treatment reached this stage, so it was a nice surprise. And also diatom, they vary in quality between species, and here we could see that Ketoceros tenuissimus did better in some of the results than Ketoceros calcitrans, which is the one used today in the archery. So that was also cool. And they also say that the faster they develop that and they reach the rudiments, it's it shows a good nutritional quality. And here our algae was the one that supported the fastest development. That's also a good sign. And yes, all of the data we had was comparable with the literature in terms of size. So our larvae were in a good range of. And for the future perspective, so one of the biggest limitations of the study was that we were not able to do the lipid analysis on site. We had to send the sample to another lab, so they are not done yet. And for the ingestion rates, we have some bias, so we are still trying to find a way to analyze it without this bias. But in the future, they want to integrate this algae to the hatchery protocol of Stellama to see how it's going on a bigger sample with like the daily production they use to replenish the stock, but not the yearly production. And also to see what happens after metamorphosis, because we stopped before. And yes, they have ongoing projects, so that's one of them, like look what happened after, and also focus more on the algae as they have a phytoplankton team to look at more of these qualities. So in conclusion, yes, our algae did well, so that was cool. It reached the objective, and it's really also give us nice things to like it gave us good things to add to the Aquabios and Stellamari objective, because they wanted to see if this algae could work, and it's very local and more sustainable. So I mean, we also show that, and if with all the lipids analysis and the ongoing one, it's shown to be really like a good algae that could be the first to like future local resource. Like maybe people will be more up to use local resources. I would like to say thank you to Yana and Amélie for supporting me during the whole project. Also to the Stellamari research platform for helping me build all the tanks and everything that was a fun moment. To the Aquabios project for founding the project, and the Marius team for enabling me to go there, and for you to listening to me today.
Keila, would you like to open?
I'm trying to think about which questions I want to ask. So.
Maybe Paola.
Je vous entends plus très bien. Tu as dit Paola?
Oui, Paola.
Okay, sorry about that. It was not clear for me. Okay, thank you very much. Again, a very nice presentation. It's exactly what we expect. So thank you so much, Lisa. I really enjoyed the introduction because you really bring us to the research question in a very, very good way. So congratulations about that. The material as a matter is also very well described. However, I have a question about the origin of the strains and
Yes.
how you cultivated the different strains, and then if this in the methodology that you have done cannot influence the results.
So the strain, yes, it was isolated maybe in 2016 from the lagoon, as I mentioned, by another researcher. And they, like in the lab, they have different teams, and I was with Viviana, which was working on the valorization of the resource. But there was a team specialized in the cultivation of the phytoplankton, like all the algae and everything, and they were providing us the algae every day. With like we gave them like we want that number of algae, and they were giving us the volume so we could fit the tanks.
Yeah.
But I don't
was
exactly.
Thinking, I understand, but I was thinking about, especially on your first result, about the variability on the data that you've got. And we know that the algal strain has their own microbiome, for example, their own capacity to grow. I don't know if the same culture medium is used for all the algae. And so I was thinking about if this variability could
maybe
be the source.
Yeah. We observed a lot of variability. That's why it's not the strongest result, but we use it to be sure that our larvae, they were not dying, but it was not the main point. Like at first we did that just to be sure that everything was going well, but that's true, we didn't take into account the variability due to the algae.
And so so it's more a question about your perspectives. If you want to have something that is more robust, on which part of your protocol you will play?
I think it's maybe C, because right now they have their diets they've been using for years and it works, but maybe we saw that maybe see how the algae of interest really influence the larvae to be sure that it's really better for it before changing everything. But I don't exactly know where in the protocol for the hatchery, at least. I don't know where they go.
Okay, so that means that the protocol is perfect.
I mean, the one they used, and it's going well. They have been using. They don't really have problem. It would just maybe be a plus if the algae is better for the searching. Maybe it's just they will have stronger juveniles.
And I could understand that you know the composite, the main compounds, the main metabolites, the main product that we can found in the different algae and the differences. It's something that it's fixed, or it's more from the literature that you think that these strains will contain more vitamins, or I don't know.
No, it's more thanks to literature. They tend to say that diatoms, they are rich in EPA and DHA, but it depends on the strain. And as we haven't looked at the profile for this one, that's why it's like we saw just physically that it did well, but we don't know exactly if it's because of the nutritional profile. That's why we really need to get the lipid. But yes, maybe they have like better contents due to like the resilience they have to show in the lagoon to be sure to survive to harsh conditions.
Okay, thank you.
Thank you very much.
Keila.
You're ready.
Aline is ready anyway.
So it was on your perspectives, you're saying that they were interested in how the juveniles after metamorphosis were doing. Do you have any data on that? Did you count how many of your samples were able to actually metamorphose?
We tried to make them do metamorphosis, but it wasn't a success. Maybe because we had a too high density, according to the technician, the high technician of the lab that was saying maybe you put too much larvae in your little thing. So that's additional stuff.
How having many larvae?
I think maybe because of the oxygen in the water and everything, the conditions were not that good.
So in your have an aquarium or
We had tanks.
tanks?
At the end, we had to empty the tanks, recover all the larvae, and then from this larvae, we took some and we put them in a little glass thing. But it also, the metamorphosis is not only because the faster they reach the metamorphosis, the more reserve they have. So it's like good condition, but it's also depend on which substrates they choose to metamorphose. It's not like that can also influence their.
It's something that is it's something that happens in the labs. It's something
Yes, they do it for their, with the normal protocol. They were trying to see also with different type of algae, but that's something they should they're going to do to see. I mean, if we feed them with this algae, it's still possible to make them metamorphose.
Okay, thank you. You can be algae if you want.
Yeah. Go with your idea.
Hello, Lisa. Thanks for this talk. So first of all, are the algae toxic for human?
Not that I know. No, I don't think they are toxic for humans.
Did they check that?
No, I haven't. But we were maybe I don't know at too much concentration. But we were using touching them and by hand and nothing happened.
That's a drastic crash test, but yeah, okay. Next question is, okay, your first set of data, when you are working with the monoalgae diet, you find that for some algae, you have a high mortality rate, while you have, when you look at the growth rate, the growth rate, okay. So for me, okay, I don't understand this data, and how can you explain these differences? Because often what happens is that when you fed the larvae, they often die because they don't grow. I mean, it's because they don't catch the prey. But here, they catch it, but still there is still some mortality. So how can you explain all this data?
I think in some tanks, maybe there were contaminations on something, because in some tanks we saw that high mortality happened, and not because we had 3 replicates, and some of maybe one replicate did super bad and the other 2 were doing well. So I think it's maybe more something that happened in one tank than I think it's maybe more variability that was due to how we were manipulating the tanks and the room more than the.
How can you test that?
Maybe looking at the water, because we were not changing the water, completely the water during the whole, or also, I guess if it's due to us.
How long was the length of the experiment?
Between 14 and 20 days.
And during 20 days, you never changed the water?
We put 40% renew every day, but we don't change the whole water. So at the end it's not the exact same, but
okay.
We don't
change. It's the environment. Okay. So okay, you explained this mortality then by not biologically, but by issue in terms of manipulation. So
maybe.
To go what Paola asked you, so what can you improve in your protocol then?
Yeah, maybe be more careful on how we are handling all the algae, and maybe because we were just putting algae in the water, maybe do not touch too much the water as well. Because we also had filters, not to be sure that the larvae, they were not going away. Sometimes we had to remove them, wash them. So maybe, I don't know, at some point we had to put something against the hole, so the water was not going away. So maybe contamination happens. So maybe, I don't know, use a really clean material. But yeah, I think it's more because of us than the condition, because the larvae, they were going from the same batch. So there is no reason that they just die in one tank and not on the other.
Okay. And how did you control the amount of food you give it to the larvae?
We calculated biovolumes, and it was the same amounts every day. And so we were giving this biovolume to the algae team, and they were giving us the samples, and we just like measured and put the right amount every day in each tank.
Okay, and are some algae bigger than other?
Yes, they were.
In terms of species?
Yes, I think some species were bigger. I don't remember exactly which ones, but it's because the larvae, they are growing, and when they are small, they tend to feed more on smaller larvae than when they grow. They will go on to bigger ones.
Of course. So meaning that, can this explain the mortality rate you have in the first graph?
Maybe, yes, maybe for the... Maybe they die at the beginning because some can feed, and then they don't die anymore because they are strong enough to feed on what's left.
Yeah, you should check that because it can explain some
why they died.
Okay, so if you have to redo your internship, then what would you change?
I would ask Mika to order the tanks earlier so we could do the whole experiment and compare like really mono and algal diets to be sure, because that's one of the limitations. We can compare them. That would have been interesting, maybe just to have an idea of the difference in size and everything, but we can't. So maybe yes, do everything at the same time.
Okay. Do you know if mixing different algae species, may lead to the secretion of some toxin or some stuff like that can kill your animal?
Not that I know. In literature, they say that mixed diets, usually they have like a better survival because of the size sometimes or the property of the algae. They just have a better feeding, but I don't think not that I know that they were toxic combination.
Okay, thank you. I'm fine.
Thank you very much.
Sorry, I have a complementary question to this point. When you put the two algae in contact with the larvae and the urchin, that will, and it take 20 days, you say? So the algae continues to grow during these 20 days?
No, we fed them every day.
You fed them adding. But from adding to two isolated sources of algae.
Yes, they were different strains, and we had like different.
So there is no issue about competition between the two algae.
No.
Okay.
We were supposed to eat everything, and then the day after we were adding new algae.
Yeah, opening across a question. Oh my God, yeah. Jacob was the first one to raise it.
We talked about the origin of the algae, and then I wondered where the sea urchins came from, because in one of the pictures you can see that they have different colors. So I wondered whether it's just the phenotype that differs or whether it's also a mix of different sea urchins.
Yes, the sea urchins, like we had to take all the we created the larvae with algae they came from Albo here. So divers they took sea urchins from the wild. We opened them and took all the gametes, and then we did the fecundation and had all like this. So they were coming from Corsica, from around there.
So it's potentially a mix of species, or is it just one species?
No, it's one species, but yet maybe different population. Like divers, they were just going there and picking them from the wild. But it was 10. We took the 10 female and 10 male that produced the most, and we mixed everything together. So it's not just one or two. We had 10 different.
Thank you.
Sophia can have a second question because last time I missed you.
Yeah, I have a question about the repopulation side of the project, because the population is declining due to fishing pressures, but there are other species of sea urchins in Corsica,
yes,
and Isn't the repopulation then more of a threat for coastal ecosystem because like a big problem in the Mediterranean is overgrazing?
Yes. Yeah, no, that's true. That if you put too much, you have the opposite problem, like you have no algae anymore. Yes, that's a good question. I don't know if, like, there are specific sites where they try to put them, so maybe where they have the higher pressures, but it's like a project that have been going for years, and like they put a lot, but I don't think a lot survive in the end. So maybe then, but it's also, yes, we take also searching to do all the lab, like what we do in the lab. So good to put them back. But yes, maybe they will have too much.
Regarding the feeding, you said that you feed them every day, and also you add more water every day, right? Did it ever happen that not all of the algae was consumed? Was there a way to know that? And was there any type of contamination of different algae when you feed the other one?
No, that's true. We didn't really look at we were just adding more every day, and we were seeing on the filter that sometimes like the filter will be going green. So of course not everything was maybe consumed, but we were like the quantities were calculated, so we don't add too much, not to have like a eutrophication in the yes. But for contamination, I don't know.
Thank you.
Yeah.
Last question.
The aquaculture part. So there's the repopulation and they put them out in the wild. But for the aquaculture, would that be on land?
Yes, I think it will be on land, but it's like the first step, the hard step is that the larvae problem, but then it takes long, like once they are juvenile, it takes like around 2 years for them to be a bit bigger. So, I mean, that's another problem that will take super long to have like aquaculture of zooplankton. It's not like fish, like it takes more time.
Cool.
So I'm going.
On this perspective, yes, we'll conclude here for this morning. Bravo. Coming back this afternoon with session 3, Alex, Laura, and Gabriela. Castanio, we will come back at 14:10. So enjoy this lunch break.
I send it.